Today, we're going to talk about photosynthesis. The interesting thing about biology is we are often very animal-centered, and I think it's because we just like things that move that are like us, and unfortunately, botany and plants kind of get ignored in general biology, but I actually think this is a fantastic topic for biology, and I really didn't understand this process until I was in your shoes. I took a majors level biology once I got to BYU, and we had a really interesting way they taught it. They actually had three different instructors, three different professors. One was a zoologist, and one, actually a pair of professors that team taught it were microbiologists, and then we had a botanist. And the botanist went and taught us all photosynthesis. And for me, it was really the first time that I understood exactly this magical process about how plants can take carbon dioxide and water and generate sugar, and to me that was sort of a pivotal moment in my biological education. So I've been very, very adamant and a champion of sort of sharing what I learned that day, way back in the in the 1980s. So we're going to talk a little bit about this process. I'm sorry, Thomas, I didn't even see you there. There I am. Okay. Now, so far I've had you learn just a couple of formulae in class. We learned how to make carbonic acid. We've learned aerobic cellular respiration. And here's the third one that you need to know. And if you look at it, you might realize that, hey, that looks a lot like aerobic cellular respiration, but it goes in the opposite direction. In fact, we find that these two reactions, aerobic cellular respiration and photosynthesis, are often described as coupled reactions, that the products of one become the reactants for the other. And for photosynthesis, basically this is building with light. Photo refers to light. And in this case, we take a gas, carbon dioxide from the atmosphere, with a little bit of water, use some light energy to combine these to make sugar and oxygen as a waste product. This was a revolution that took place in the history of the world. Photosynthesis, go ahead. Photosynthesis wasn't original. It shows up probably about 2.1 billion years ago. So we have life for about 1 billion years before that. And photosynthesis, this remarkable ability to make food, was mind-blowing. And the interesting thing about it, it completely changed the atmosphere. And if you think about oxygen, oxygen is incredibly reactive. And for a long time, it's considered to be a poison. And all of a sudden, we have free oxygen in the atmosphere, and it was because of the photosynthesis that was taking place by bacteria. So it sort of helped create the world that we live in, but it sort of started out as a waste product, this little CO2, a little oxygen there. Now, if we look at a plant, plants are usually green in color. There's a reason for that, and we'll talk a little bit about that. But if you look at a typical vascular plant, you'll notice that the main organs of photosynthesis are going to be the leaves. This is where most of the photosynthesis takes place, but any part of a green plant that's green is generally going to be photosynthesizing. So we usually think about the leaves of having the greatest amount of photosynthesis happening. And if we look at the structure of a leaf, we'll find that in the middle of the leaf, what we call the mesophyll, has the densest concentration of cells that have the chloroplast organelle. Now, if you look at a typical leaf, you have a little waxy cuticle on top. For a vascular plant that's terrestrial, drying out is a big issue. So, great way to protect against water loss is by having a layer of wax, all right, nonpolar wax on the surface. If you go to a really arid place, I grew up in Southern California, which is pretty dry, a lot of the plants there, you can actually scrape the waxy cuticle off because it's so thick. Arid places often have leaves of plants that are pretty well protected. You'll also notice here that we have these beautiful little openings. These are called stomata. That just means mouths in Greek. And it's a way for carbon dioxide to go in for photosynthesis and CO2 to actually exit. And usually they're found on the underside of the leaf. And as you can see, they also have these special guard cells. These guard cells are filled with water. And when that water dries up, when we have a little bit of desiccation or a dry, hot day, that will cause that to close. When they fill up with water, that means it's nice and humid. That'll open up and allow the gas exchange to take place. And most of that gas exchange takes place on wet, humid days or at night. Now, we also have in the middle, the very middle of the leaf, we have these beautiful cells. This just means mesophyll, which means middle of the leaf. And this has the highest concentration of chloroplasts inside the cells. And if you remember when we talked about chloroplasts, they're a unique organelle in that they have two membranes. They have an outer membrane and an inner membrane. We just have a couple of organelles that are like that, the nuclear envelope of the mitochondrion. And we know about the prokaryotic heritage of these chloroplasts. It's sort of a cell within a cell. That's how a good way to think about a chloroplast. Now, if you remember, inside the chloroplast, in the interior of the chloroplast, is this complicated membrane system. We have these little green discs, and these are called thylakoids. So each little individual little disc here is called a thylakoid. The thylakoids are arranged in stacks. In fact, when I look at them, it makes me hungry for Thin Mints. If you're a Girl Scout cookie fan, I just think about a sleeve of Thin Mints there. All right? And that little column is what we call a granum. Right? And then it is surrounded by a fluid-filled space called the stroma. Yes?
Wax layer, if you take that wax layer off, does it grow back?
It probably will. It's the epidermal cells that secrete it. So it's secreted by those top cells. All right. Now, there's actually some importance to these different structures, and I want to mention one. Photosynthesis is composed of two stages. One stage called the light reaction, and one stage called the dark reaction. The light reaction occurs within the thylakoid. The thylakoid is like a little sac. It has a fluid-filled space that we call the thylakoid space. It also has the pigment chlorophyll. So chlorophyll is found within the thylakoid membrane, as we call it. So this is our thylakoid membrane. All right. So these is chlorophyll. Basically what happens here is that light energy is converted into chemical energy. And we'll find out that that chemical energy will come about because it'll be in the form of ATP and some energized electrons in a molecule called NADPH. Right, so that's why the thylakoid is important, because this is actually where the light energy is harvested, and that energy is going to be stored as in the bonds of ATP and in electrons stored in NADPH. Now the dark reaction takes place in that fluid-filled space outside in the stroma. And what it does is it takes carbon dioxide and converts it into glucose with the help of these two sources of energy. So it's kind of a two-step process. Light energy is captured to make chemical energy. That chemical energy is used to convert carbon dioxide into glucose. And this takes place in that fluid-filled space called the stroma. That's sort of the big picture. Right? So when we learn about these little parts, this is what's going on inside those parts, and we'll talk about them in more detail in just a moment. This was the part that amazed me when I was an undergraduate, was this part right here. How in the world do we take electromagnetic radiation and convert it into chemical energy? And so that's what we're going to talk a little bit about today, this particular process. As you know, light comes in a variety of different forms. And light is classified according to its wavelength. Right? So we have light that is in very, very short waves that we call gamma rays. That's the shortest ones. It takes a lot of energy to make a gamma ray. This is why we find gamma rays showing up in supernovae or when black holes merge. All right, so we can actually detect the presence of gamma rays and we go, oh, okay, something really big is happening out there. The next big, X-rays, which are a little bit longer but still super powerful. Ultraviolet radiation, all right, which we know comes from the sun. And then what we call visible light. All right. Beyond visible light, we have light that's in longer wavelengths. This is our infrared. A little bit longer than that is microwaves. A little bit longer than that is radio waves. And some radio waves are so long, the wavelengths are measured in meters and kilometers and tens of thousands of kilometers. Right? So light, it spans the spectrum by the length of the waves. Now, if we look at visible light, which is just a tiny, tiny fraction of the entire electromagnetic spectrum, we find that it ranges in wavelength between about 400 nanometers and 700 nanometers. All right? A nanometer is a millionth of a meter. Right? So, pretty dang small. Now, one thing that we've learned in physics is that light has a inverse relationship with energy. So the shorter the wavelength, the more power that light has, the stronger the photons. For example, gamma rays, which are really, really short, have the most amount of energy. And radio waves, which are really long, have the least amount of energy. All right, we saw that with mutagens. You know, there's a reason why ultraviolet light damages DNA. That short wavelength photons have enough energy to actually damage DNA, alter biological molecules. All right? And X-rays, as you know, even stronger. Visible light is powerful enough, all right, to modify biological molecules, but not destroy them, not break them apart. And that's why this is the visible wavelength we can see. It's because this is the little sweet spot where biological molecules aren't going to be destroyed by them, but they are going to be altered by them. Okay? And this range is between 400 and 700. And if you notice, the different colors of the rainbow are all produced by different wavelengths of light. So if you learned Roy G. Biv as a kid, all right, 400, all right, violet light, all right, this is going to have a lot more energy than 700. So blue light and violet light have more energy than, let's say, green or orange or red. All right? So that has the greatest amount of energy. Red over here at 700 will have the least amount of energy. Now, we live in a world where we see a lot of color. And colors are created by pigments. How pigments work is that pigments are often very selective on the wavelengths of light that they absorb. If, for example, we look at a white sheet of paper, when you look at a white sheet of paper, all right, the reason that it's white is that all the wavelengths of light, of visible light that are striking this, are not being absorbed. They're being reflected back. The colors that you see are the wavelengths of light that are not being absorbed by the pigment or the substrate. It's the wavelengths that are being reflected back. So if you go and look at something that has, for example, a black color, right, that means all the wavelengths of visible light are striking this and not being reflected back. So it looks black to us. When we see something that has a color, for example, my blue shirt, this is absorbing all the wavelengths of visible light except for blue, which is being reflected back. When we look at yellow, all the wavelengths of light except yellow are being absorbed by the pigment, and those are being reflected back. Now, we live in a very green world, and the dominant pigment for photosynthesis is one called chlorophyll. This literally translates as green, chloro means green, leaf. The green leaf pigment. And this is what chlorophyll looks like. Now, if you're wondering about that, you go, that's kind of funky. How would we classify this one? Turns out this is classified in the lipids. It's classified as a terpene, a little strange little group of lipids called a terpene. And this is the dominant pigment on the planet. This is what gives us our green world. Now, there are several different versions of chlorophyll. The dominant pigment is called chlorophyll A. Chlorophyll A. There's another one called chlorophyll B, which is, we call it accessory pigment. And you can see the big differences between a methyl group and a carbon with a hydroxyl group there. Now, right now, during the fall, when we see the leaves change, right, we're seeing all these other different colored pigments that we usually don't see in leaves. So when you look at the autumn leaves and see the browns and the reds and the yellows and the oranges, turns out those pigments were already there. They've been there all along, but they've just been masked by chlorophyll. Right? During the fall of the year, a lot of plants go and turn off their chlorophyll. So they stop making chlorophyll. And so what you see is the pigments that were masked by the dominant chlorophyll A. Now, these other pigments also play a role. They also are involved in photosynthesis, and we'll see what they do in just a bit. But we have a name for these. If chlorophyll A is our dominant pigment, the other ones are called accessory pigments. There's some beautiful other ones. We've seen chlorophyll B. There's also a chlorophyll C. Right. Oftentimes when you go and you take a look at a carrot or corn or a lovely yellow squash, you're looking at a pigment that's called a carotenoid. In fact, beta-carotene is a great example of a carotenoid. There are other accessory pigments too. There are ones that produce a yellow, a red color or a purple color. Right. These are called phycobilins. All these different pigments that we find in leaves actually play a really important role because they absorb different wavelengths of light. Wavelengths of light that are ignored or reflected back by chlorophyll A. Reflected back by chlorophyll A. Now what I'm going to show you is what we call an absorption spectrum. How an absorption spectrum works is we have a beautiful machine in science, and it's called a spectrophotometer. Spectrophotometer. A spectrophotometer is this nifty machine where we can actually go and take a pigment. We put it in a little glass test tube called a cuvette and put it inside our little machine. And then we shine a light through it. And we see how much light is transmitted through it on the other side. And the cool thing about the machine is we can do it one wavelength at a time. So we can start it at 400, go to 401, 402, 403, all the way to 700. And we can see how much light goes through at different wavelengths. And then plot a graph to see how much absorption. Because pigments are very specific on the wavelengths of light that they absorb. So you can do that with all these different pigments, and you'll get a different absorption spectrum. So I'm going to show you this absorption spectrum that we do with a spectrophotometer. So this is what it looks like. We're going to begin with our dominant one. This is chlorophyll A. So if we stuck chlorophyll A in our spectrophotometer and started at about 400 here and we go clear to 700, you'll notice that it has two spikes. Here it is, its most absorption. This is where it has its greatest absorption. And you can see that's about 425. And then it drops off very quickly and then really has no absorption until we get here to kind of the yellow. And then it peaks again here at the red. So it has two peaks, one here in the violet-blue range, one here in the sort of orange-red range. Now the reason that chlorophyll A looks green to us is because if you look around here, there's no absorption. It's reflecting back that light. Now if plants just had that, if that was the only pigment they had in their leaves, they would miss out on all this energy. So this is why they have multiple pigments. That way they can absorb light energy from every visible wavelength. So if you look, for example, at chlorophyll B, which is right here, you can see, wow, that absorbs greatest here at about blue-green region. So in this case, this extends the absorption spectrum for the plant. If we look, for example, at beta-carotene, one of our carotenoids, you know, the reason that it's sort of yellow or orange or red is because it doesn't absorb any wavelengths over on this side, but absorbs everything on this side. So that's why it looks, you know, yellow or orange or red to us. Here's one of my favorite ones. This is phycoerythrin. As you can see, that means red, all right? And as you can see, it has some absorption here in green and yellow, a little bit in the blue and violet, but absolutely none here. So this is a plant pigment that would make things look red. Here's one that would make things look blue. As you can see, it has its greatest absorption over here in the lower part, in the green, yellow, and red, and then pretty much not much at all at blue, so it's going to look blue. Right? In fact, that's what cyanin means. It means blue protein. Okay. So that's what these different pigments can do. And it allows the plant to photosynthesize through a whole variety of different wavelengths. Now, what we can do next is do the same thing with our spectrophotometer. Well, actually, on this way, we can do something a little bit different. We can take our spectrophotometer, all right, and we can show a wavelength of light onto a green leaf. And then we can go and measure the photosynthesis that takes place when we expose that to a specific wavelength. Now, how do we measure photosynthesis? It turns out it's really simple. One of the products of photosynthesis is oxygen gas. So you can measure the amount of oxygen that's being given off by a green leaf. That tells you the amount of photosynthesis. The more O2 given off, the greater the photosynthesis. Right? So this is the same spectrum, but again, we're just looking at one particular plant and one particular leaf. This comes from Anacris, which is a type of aquarium plant, right? Sometimes called Elodea. Right? And as you can see, here is our rate of photosynthesis. That's the amount of oxygen being given off. And you'll notice that it matches up fairly well with the dominant pigment, chlorophyll A. So we have two spikes, one here in sort of the violet-blue range, and then a second spike over here in sort of the orange-red range. But you'll notice that because of the other pigments, if we didn't have any other pigments, this would probably just go like this. All right, we would have nothing here. But you'll notice that because of the other pigments, all right, this plant can actually go and photosynthesize in these middle wavelengths, something that it wouldn't be able to do with just chlorophyll A. So those accessory pigments allow the plant to use all the wavelengths of light in the visible spectrum. All right. Any questions on those different pigments? Okay. Now, one really cool thing that happens with electrons is that As you know, electrons have different energies. Those electrons that are closest to the nucleus in that first energy shell have the least amount of energy. Those that are farther out in the more valent shells are going to have greater amount of energy because they are withstanding that electrostatic attraction. The nucleus is positive, electrons are negative. Those that are closest, all right, are the ones that don't have the energy to be far away. All right? So it's almost like a spring. The more you pull it out, the more energy you got. All right? The ones that are farther away have the most energy. Now, it turns out that pigments, how pigments work, is that they will absorb a photon at a certain wavelength. Turns out these pigments are very, very specific on the small little range of wavelength that will actually go and cause their electrons to be bumped to a higher energy level. Let me show you a little diagram here. So if we have, for example, a pigment, let's say it's chlorophyll A. So we shine a wavelength of light, let's say that it's about 425. So we have a photon at 425 nanometers. And what happens is that wavelength is just the right size to stimulate that electron to move to a higher energy level. So we say that that pigment, that molecule, that electron has been photoexcited. Has been photoexcited. So as you can see, it bumps it up. That energy from that photon has now been stored in that electron. It's potential energy. It's almost like taking water from below a dam and moving up to the reservoir above the dam. Right? Now we have stored energy. Right? So this is what we call photoexcitation. That's how pigments work. Right? That's how pigments work. They absorb that energy and it boosts their electrons to a higher energy level. Alrighty. Now, how long does it stay up there? Well, it turns out that this is usually very short-lived. Most of the time what happens is when an electron is boosted to a higher energy level, it just stays at that higher energy level for a fraction of a second. Just for a fraction of a second. And then you'll see that it drops back down. It'll stay up there for a bit, and then it'll drop back down. It took energy from light to make it go to the next one. But it's also going to release energy when it drops back down. So, when it drops back down, it gives off that amount of energy in two different ways. One way is by light. So, when a photon goes and strikes a pigment, and it goes and causes that to be energized, and it stays there a fraction of a second, when it drops back down, sometimes that energy is being released as light. Oftentimes when it does that, it will be at a wavelength that is longer than it was originally. Or, it gives off the energy as heat. So, let's talk about the light for a second. What you're seeing there is chlorophyll. This is chlorophyll A in the Erlenmeyer flask. And what we're doing is we're exposing it to ultraviolet light. And you'll notice you go, well, I thought chlorophyll A was green. Why is it showing red here? what you're seeing is a thing called fluorescence. Fluorescence happens with a lot of substances. When light of a certain wavelength goes and strikes a pigment and causes photoexcitation, and those electrons go to a higher energy level, it turns out that when they drop back down and they give off that energy, they give it off as energy in a longer wavelength. The light they give off has a longer wavelength. So in this case, the light being given off has a longer wavelength than the ultraviolet. You'll notice it's red in color. Ultraviolet is like 350. That's 700. That's what we mean by fluorescence. That's what we mean by fluorescence. Turns out that's how these lights work as well. Right? These little fluorescent lights, as we call them, work in exactly the same way. The little things are what's happening is that inside those tubes we have ultraviolet light. This is light we cannot see. And it strikes, if you've ever looked at a fluorescent bulb and you see that white coating, and you've wondered, what the heck is that white coating? That's phosphor. And what happens is when a photon of ultraviolet light strikes that, right, that phosphor absorbs it. The electrons get boosted to a higher energy level and they quickly drop. And they give off their energy in the form of light. But it's a longer wavelength. In this case, it's the wavelengths of visible light. So you're seeing fluorescence right there. That's the same process that we see here with that. Some of my favorites are fluorescent minerals. You can actually go and take minerals like calcite, all right, wolfenite, all right, all kinds of cool minerals, and you expose them to ultraviolet light and they will glow different colors. They'll glow greens and yellows and oranges and blues and stuff, and it's all this same fluorescence. The other important one is that we often go and get heat coming off. This explains why a car that is dark in color gets much hotter on a summer day than one that's white. Okay? If you think about what's happening with let's say a black colored car, as that sunlight goes and strikes that pigment, it's absorbing all those wavelengths of light, and those electrons are being boosted to a higher energy level. They stay there for a fraction of a second, they drop back down, and they give off that as heat. Now, if we think about a white car, that light's being reflected back. All right? It's unpigmented. So that's why dark substances are a lot more warmer when exposed to heat and light than what we have with something that's light colored. And that's because of what the pigment's doing and what the electrons are doing. Okay. Any questions about what light does with that? Okay. All right. Now, I want to come back here to the thylakoid membrane. It turns out that there is not an equal distribution of chlorophyll. We kind of get this impression that the membrane is just solid chlorophyll. It turns out that it's not. The chlorophyll are in little islands, little clusters of chlorophyll molecules, about 300 chlorophyll molecules per island. And these islands are scattered throughout the membrane. So there are spaces between the little islands of chlorophyll molecules. We have a name for these islands. These islands of chlorophyll molecules in the membrane are called photosystems. So imagine distributed through the thylakoid membrane are little islands, little clusters of chlorophyll A molecules. Right? And this is what we call a photosystem. In the middle of these photosystems is a special little pair of chlorophyll molecules called a reaction center. You see the little reaction center right there, the one that's represented by a square? Right. This is pretty much the same as these, but they're kind of special. Right? It's a pair of chlorophyll molecules that will play a central role in converting light energy into chemical energy. Now, how a photosystem works is pretty basic. A photon comes in, it penetrates the leaf, let's say gets to the mesophyll, hits a plant cell, goes down, hits a chloroplast, hits a thylakoid, all right, and strikes one of these photosystems. So here it is, striking one of our little chlorophyll molecules. Right? Now, what happens is that chlorophyll molecule, it absorbs that photon, and its electrons get boosted to a higher energy level. Right? Stays like that for a split second and then drops back down. Now, in a process we don't quite understand, in fact, we think that this might occur at the quantum level, I mean really, really tiny level, right? That energy that was absorbed and released by this gets picked up by the neighbor, and its electrons get boosted, it drops back down, and then that energy gets absorbed by the neighbor. So that energy bounces from one chlorophyll molecule to the next. So it keeps doing that, bouncing around, until it gets to this special reaction center. Now, the reaction center has two special electrons. All right, so they get absorbed, they absorb that energy, they get boosted to a higher energy level, and rather than going and dropping back down and keeping the process going, those electrons are promptly stolen. They're stolen by a molecule. And they're called a primary electron acceptor. So, here's the same little picture. This is our thylakoid membrane. So here's the phospholipid bilayer. Here's our little photosystem. And inside the photosystem, we have all these little, about 300 of these chlorophyll molecules. The light comes in, boosts the energy level of the electrons that gets passed from one to another until we get to the reaction center, and that's our reaction center. Once they receive that energy, they boost their electrons to a higher energy level, and then they're promptly stolen by a molecule called the primary electron acceptor. So now the primary electron acceptor is holding two excited electrons. Electrons that are at a higher energy level. They're much higher than what we have with the ground state. Now, this is what a photosystem looks like. You can see that it's some pretty wild proteins, transmembrane proteins, and if you look real close, you can see, oh, look at all the chlorophyll. That's all chlorophyll A embedded in there. And this is all taking place right here in the thylakoid membrane. Okay. Any questions on how a photosystem works? Okay. Now we're going to go and look at how the light reaction works. Remember, the big thing about the light reaction is we're going to take this light energy and convert it into chemical energy. Now, we call this the light reaction. The light reaction is light dependent. It has to occur. It can only occur when it is exposed to light. So sometimes we call this the light dependent reaction. Okay. Turns out that this is all going to take place right here, and it's going to involve two photosystems. So we're going to start with the first photosystem. Now, there's two photosystems. The first one is called Photosystem I, and the second one, guess what? Photosystem II. They're named in the order of their discovery. Right. So let's take a look at Photosystem I. All right. Now, if you look at Photosystem I, right, it looks just like the photosystem I just described, and you'd be exactly right. In fact, Photosystem II looks just like this too. We've got this beautiful little cluster of chlorophyll A molecules. Right. So, light energy from the sun, right, it bounces around. So these little chlorophyll molecules, their electrons get excited, they get boosted, they drop down, they pass their energy to the next one, to the next one, to the next one, the next one, until they hopscotch to our reaction center. The reaction center, as you know, the electrons get boosted to a higher energy level, and then they get promptly stolen. They get stolen by a primary electron acceptor. And here's our two energized electrons now being held by this molecule here. Now, this molecule here, called the primary electron acceptor, doesn't hold on to these energized electrons very long. It passes it to a middleman. This FD is an iron-containing molecule called ferrodoxin. Ferrodoxin. In fact, I'll write that up just so you can impress your friends. Ferrodoxin. And then we have an enzyme called NADP reductase. Right? Oftentimes, enzymes tell us what they do. If you remember, when we talked about cellular respiration, we talked about oxidation and reduction. Right? Reduction is when a substance gains electrons. So in this case, NADP actually goes and picks up those two electrons plus a hydrogen and becomes NADPH. Now you might be wondering, well, what the heck is NADPH? Is it anything like NAD? And you would be right. This is very similar to what we talked about with aerobic cellular respiration. Right? NADP stands for nicotinamide, adenine, if I could spell today, adenine dinucleotide phosphate. This is that nucleic acid. Very similar to what we find with NAD in aerobic cellular respiration. And we know that just like in aerobic cellular respiration, it likes to carry energized electrons. So NADP, as you can see, grabs 2 electrons plus a hydrogen and gives us NADPH. And that NADPH is going to make its way to the stroma. That's one of the chemical energy molecules that's going to allow us to convert carbon dioxide to sugar. So very similar to what we saw with NAD in aerobic cellular respiration. Now, I want to mention one more thing, and that's this strange name. You see the P700 up there? The P700 right here? That is the technical name for the reaction center, which is right here, of photosystem I. The reaction center of photosystem I is called P700. P700. Now, the 700 actually represents the wavelength of light, where that electrons are most sensitive. So they have their strongest absorption at 700 nanometers. Hence the name P700. The strongest absorption at 700 nanometers. That's right there in the red range. Right there in the red range. So P700. Now, we have a problem. What is P700 missing now? Now that the electrons are gone? Electrons. We have to replace them for this to keep going, to keep working. So we've lost 2 electrons. We're going to have to replace those so we can do this again. And that's where photosystem II comes from. Photosystem II will replace those 2 lost electrons. Will replace those 2 lost electrons. Right? So one of the functions of photosystem II is to replace the 2 electrons lost by the reaction center of photosystem I. Any questions so far? Alrighty. So, let's take a look at photosystem II. And you'll look at it and you go, oh, that looks just like photosystem I. It's got the same little system here. We got a primary electron acceptor, all that good stuff. All right. So, let's see how this one works. Very similar to what we saw with a typical photosystem. Light energy. Our chlorophyll A absorbs the wavelengths of light. It boosts the electrons of the chlorophyll A pigment molecules. Those electrons get boosted to a high energy level. They drop back down. They pass their energy to the adjacent ones. And they keep bouncing from one chlorophyll molecule to the next until we get to our reaction center. Now you'll notice our reaction center has a different name. It's called P680. P680 is the name of the reaction center in photosystem II. And we call it that because, like the other one, that is the wavelength of light that those chlorophyll molecules have their greatest absorption. Right? That's the one that they have the greatest absorption at 680 nanometers. So, those electrons bounce and they arrive at our reaction center. That reaction center, as you know, has two chlorophyll pigments, molecules. Those electrons get boosted to a higher energy level, and then just like in photosystem I, they get stolen. They get stolen by a primary electron acceptor, and there it is. Now, something really weird happens now. You might see something that looks familiar. Where did we see cytochrome before? Think about the inner membrane of the mitochondrion. Where did you see a cytochrome? What stage of aerobic cellular respiration uses cytochrome? Electron transport chain. Just like the inner membrane of a mitochondrion, chloroplast also has an electron transport chain. And if you remember, at the beginning of the electron transport chain, we have energized electrons that get passed from one protein to the next. Right? And by the time they get to the end, that energy has been extracted from. Those electrons are spent, but what's the energy used for? To pump hydrogen ions into the intermembrane compartment, right? Same thing happens here. But it's not the intermembrane compartment of a mitochondrion. It's the thylakoid space of the thylakoid. So, what's happening is we have, right, this enters an electron transport chain. And what happens is as those electrons are passed from one protein to the next, hydrogen ions are pumped into the thylakoid space. Wow. And guess what? There's a channel protein that's specific for hydrogen. And what do you think's attached to it? ATP synthase, the turbine. So as the hydrogens flow through the channel protein, they spin the turbine and make ATP. It's chemiosmosis. It's exactly the same. We have an electron transport chain in the thylakoid the thylakoid membrane. We have a channel protein with an ATP synthase. As hydrogen ions move through the channel probe, it spins it and generates ATP. Chemiosmosis, an electron transport chain in a chloroplast. Wow. Wow. Isn't that wild? So that's where the ATP comes from. It's the same mechanism that we saw in aerobic cellular respiration. That's where the ATP comes from. So here's a nice little picture of it. Let's take a look at this one here. So, here's our electron transport chain. All right. The electrons get grabbed by a primary electron acceptor. This is photosystem II. Gets passed down the electron transport chain. Hydrogens are pumped in to our thylakoid space. This is active transport. Hydrogens can't get through the phospholipid bilayer because they're charged, but we have a channel protein here, and the hydrogens flow following their gradients, and we have a beautiful little ATP synthase that spins, and that flow of hydrogen ions generates ATP. And there's our ATP. So photosystem II generates ATP. Now, you'll notice that we have a problem. What was the purpose of photosystem II? Replace the electrons lost in photosystem I. So those electrons eventually make their way back to photosystem I. But now, what problem does photosystem II have? It still has 2 electrons missing. We've just stolen from Peter to pay Paul. Right. So we have to get 2 more electrons. And that electron comes from water. Photosystem II, to replace the 2 electrons that it gave photosystem I, strips the electrons from water. Now, if you think about our formula, if you remember, photosynthesis is CO2 plus water gives us sugar and oxygen. All right. In the light reaction, what happens is the water is split. Here in photosystem II, water is split, and out of that we get 2 electrons. And those 2 electrons go to replace the ones that were given up by photosystem II. So you're wondering, oh, okay, so that's where the water comes in, and it also explains where the oxygen comes from. We know that oxygen is a waste product. Here is your O2. So it's photosystem II that's actually going and producing the oxygen gas as a waste product. This is where the oxygen comes from in our atmosphere. It comes from photosystem II. Isn't that cool? So now we've knocked out 2 of the parts of our formula. Now we know what the water did, and now we know where the oxygen came from. Now we know where the ATP comes from, and now we know where the NADPH comes from. Sorry, it's so low there. 1/2 O2 plus 2H plus. Well, kinda. We had to balance the equation there for it to make sense. Isn't that cool? Isn't that cool? All right. That's awesome. And that's all taking place right here. If you think about it, think about what we just did. We converted light energy into ATP and NADPH. We converted light energy into chemical energy. All right. So here's how they're combined. All right. We saw that photosystem I, the energized electrons get stored at NADPH. They're going to go to the Calvin cycle, which is the dark reaction. And then the 2 electrons that it lost are replaced using photosystem II. All right. It enters electron transport system. This is where the ATP comes from. All right. This is going to go to the Calvin cycle. And then in order to replace those 2 electrons, we split water. And so water is used and oxygen is given up as a waste product. So we're halfway there. All right. Any questions on the light reaction? Okay. Let's look at the dark reaction. This is sometimes called the Calvin cycle. It's named for a wonderful researcher named Dr. Melvin Calvin. All right. He was helped with a colleague named Benson, Dr. Benson, so sometimes you'll see it referred to as the Calvin-Benson cycle. But this is the cycle that's actually used to take carbon dioxide and convert it into sugar. That's the big picture here. So called the dark reaction, the Calvin cycle, the Calvin-Benson cycle. Sometimes you'll see it referred to as the light independent reaction. Oftentimes when we love something, we have lots of different names for it. And biology is no different. Now, I absolutely love this next little diagram. This comes from your textbook, and it shows you the big picture of how photosynthesis works. As you can see, it tells us where this is occurring. There's our chloroplast. All right. Here is our thylakoid. This is a granum here. All right. We see how the light reaction generates ATP and the energized electrons here in NADPH. They move out to the stroma. These help power the Calvin cycle. We also saw that in the light reaction, water is split and oxygen is released as a waste product. Okay. Now, in the dark reaction, or the Calvin cycle, we're actually going to go and see how the carbon dioxide is what we call fixed. This is a gas, atmospheric carbon dioxide. We're going to turn it into a solid molecule. We're going to turn it into sugar. So we're going to fix it. All right. So this is called carbon fixation. So it's going to enter this metabolic pathway. And as you can see, it's a cycle because, as you can see, our NADP, once it's given up its electrons and hydrogen, goes back here and gets recharged. The ATP, once it's used, is recycled back to the thylakoids to get recharged as well. And as you can see, we get sugar as our final product. Now, the Calvin cycle can be a little scary. It has some fairly big names, but it's really quite easy. There's less to remember about the Calvin cycle than there is the light reaction. But it does have a couple scary names, but I'll help you out with those. You'll be lucky because we often just abbreviate them. Alrighty. Now we have three major phases of the Calvin cycle. The first phase is what we call carbon fixation. That's the first phase. Carbon fixation. So here it is. This also comes from your textbook. And here is our carbon dioxide. Now you might be wondering, what does it bind to? How does it begin? Well, it turns out that it binds to a 5-carbon carbohydrate, a 5-carbon sugar, by the lovely name ribulose. So here's ribulose. Now ribulose, in this case, as you can see, has 2 little phosphates attached to it, one at each end. So we call this ribulose bis. Bis just means 2 phosphates here, bisphosphate, one on one end, one on the other. And in this case, we like to just give it an abbreviation, RUBP. RUBP. All right. This is probably one of the most abundant carbohydrates on the planet. And you've probably never heard of it before. This is in every plant, every algae that photosynthesizes, is this little sugar right here. Now, we have an enzyme, a really cool enzyme, that goes and attaches carbon dioxide to this 5-carbon sugar. And it has a lovely name. It's called, as you can see, Rubisco. That's the enzyme. Of course, that is an acronym. Right? In fact, its full name is this. And you can impress your friends. Ribulose biphosphate. All right. That's what the Rubis stands for. The CO stands for carboxylase. You think, oh, that sounds like carbon dioxide. So it attaches carbon dioxide to ribulose. This is an amazing enzyme. Look at that beautiful protein. Gosh dang. So symmetrical, so lovely. And just to kind of show you what it looks like here, that's it. You notice that the active site, that's the active site, and you can see the amino acids. So there's a histidine and an arginine, and there's a lysine and aspartic acid. And if you remember back when we talked about proteins and you went, oh, I remember those. All right. In fact, this one, if you remember, has a positive charge. In fact, this one's basic. Remember the positive R groups? And there it is, binding to that one. All right. Here's one that has a little hydrogen. This is a hydrophilic. All right. This one's acidic. So as you can see, little positive charge there, negative charge binds to it. And you can see this is actually where the CO2 goes and binds. It's beautiful, absolutely beautiful. All right, gorgeous little active site. So that's the big function of Rubisco, to attach that carbon dioxide to our 5-carbon sugar. All right. Now, we're going to stop right there. I'll show you what happens next when we come back on Wednesday. Anybody have any questions? All righty. Well, cool. It's going to be on membranes today. Just membranes. What was that? Oh. Well, you'll be ready for after the holiday then.